Hi guys, I am here in Uppsala with Maja Malmberg, who is a researcher at the Swedish University of Agricultural Sciences.
Yes.
And
Yeah.
Are you ready?
Yes.
Let's go!
Let's go.
So, did you always know that you wanted to have a career in science?
No.
No?
No, I didn't know.
What did you know?
I just knew that I didn't know what I wanted to do.
Yeah.
But I like to study and learn new things.
Yeah.
So then I figured, okay, I just continue to study and study things that I find interesting, and hopefully I find a job later.
Yeah.
And then I kind of continued.
Yeah. But how did you then end up in the field that you're in?
Because
there's, I mean, if we talk about science, one thing is to study, but there's a lot of fields to study.
Yeah, it's true. Now I think when I was a child, I lived in Africa for 2 years, and I was influenced by like this. For example, malaria was very present there in my life as a child there. So then I was intrigued by that. So I knew I wanted to do something with infectious diseases and in Africa. So I was choosing between HIV and malaria for my degree project, and then it became malaria, and after that I've been going more into viruses. I find infectious diseases very interesting
Because of...
It's like they are super small, like this virus, for example, now that is changing the whole world. It's like when you hold your hands like this, imagine the difference if you would hold the Earth. That's how much smaller. It's like 27 million times smaller than this. So it's like the same equivalent if I would hold the whole globe and then that hold the virus.
Wow.
Something that small, like 27 million times smaller than us,
is able to
just completely change everything we know about society.
That's amazing.
Yeah. Then I got the possibility to do a postdoc where we were about to find new viruses. Yeah. So my postdoc was all about discovering new viruses, and we do that using high throughput sequencing, and bioinformatics. So basically new tools became available that made it possible to find viruses that we've never previously been able to find.
Because they're so small.
Exactly.
But what is a virus actually? Is it a life form, or...?
It's considered to not be alive.
Yeah?
So it's actually like an infectious agent that needs other cells to replicate.
So it
cannot live by itself, so to say.
So it has to always sort of like
hijack
somewhere.
Yeah.
That's cool.
And they basically exist everywhere, because all kind of life forms, all kind of cells that exist, like algae, archaea, bacteria, everything has viruses that can infect them. So
so they just hijack a cell and make it their home, or what is the purpose of a virus? Does it just want to survive and
I guess like it's evolution, so it doesn't really have a will or a purpose as such, but some take over the system and destroy it and make sure that they multiply, like this coronavirus now. It's taking over our bodies and making sure we are multiplying this coronavirus, and then we spread it and it continues. Yeah. But other viruses can stay latent. For example, like herpes viruses that can give this
cold sores.
They can stay latent for many, many, many years.
You have been working with lots of different animal species. You've been working with, I saw, dolphins.
Yes.
And you have worked with seaweed in Antarctica.
Yeah, or sponges.
And you have been working with, cattle in Uganda.
Yes, yeah.
And you've been working with swine flu.
Yes.
In Sicily.
Yes, exactly. And some other more different animals, like penguins. They are not published yet, those things, but also penguins and cats and dogs and geese and lots of work with ticks also.
Yeah. And why?
Why so many different, or why this?
Why?
When these new methods became available to find new viruses, was basically a lot of things we want to explore. Like, okay, so what viruses do these animals have, and what viruses are? So it's also important to know even like these viruses that might not cause disease.
Yeah.
What are they and what are they doing? And
to create sort of a catalog of viruses of the world.
Yes.
When you do sequencing,
yes.
How do you then know that this is a DNA sequence from a virus or a human? Do they have some sort of like specific way where they're different?
So basically mainly use like with databases. So with every sequence of every virus that has ever been published, and then we compare our data set with that existing. So if we would find a virus sequence that doesn't look anything like anything previously published, then we cannot find it, so to say.
Yeah.
But that so far hasn't happened. So usually there is some similarity with existing sequences, and then you can start guessing like, okay, since we found this in this animal and it looks like coronavirus, for example, then can start
moving around and
exactly, and describing
like a puzzle piece almost.
Yeah, it's a bit like detective work. Viruses do evolve and very different for different viruses. Like this coronavirus now that are circulating is evolving much faster than the African swine fever virus. Coronavirus is a family and they infect basically all kind of animals.
Yeah. So we actually have.
It's called.
We actually have coronavirus in our systems right now, but not the same.
Probably not. Like there are 7 coronaviruses now known to infect humans, and they usually cause normal, like a common cold.
Yeah.
So you would probably not have it now unless you had a common cold.
Okay.
And of course, if you have a common cold, it can be many different other viruses.
Yeah.
And then it's the MERS and the SARS that are the 2 like previously known coronaviruses that are deadly.
Coronaviruses, as you say, has been found in different animals, but not in humans until recently with the SARS and the MERS. There were other like these common cold viruses have been described previously. Yeah.
And it's just because they just cause common cold, no one talked about them. Like it was not. So the first time human doctors started talking about coronaviruses was when it came to SARS, so in 2002. But before that there's been a lot of discussion and research done on coronaviruses in animals because it has been causing diseases in bovine and in poultry and in pigs. So veterinary virologists know a lot about coronaviruses. The more you study virology, the more unlikely you think that this kind of event actually is, because what is required is like first the virus needs to have the ability to bind to the receptor. Like usually receptors are very specific for cell types and for different animals.
Yeah.
And the virus to enter a cell, it needs to bind. So that's like the first thing. So it needs to have the ability to bind to a specific receptor that is existing in this animal that it wants to infect.
Yeah.
But then it's the whole thing, like because it's totally dependent on the cell that it infects. So it needs to modulate all the immune system of the host, but also modulate like the cell and interact with all the components of the cell. So it's super unlikely event that this happens, in considering then that we have all these viruses everywhere all the time. This hasn't happened in a very long time.
No.
So it makes coronavirus a bit different because it has what is called recombination. So it has the ability to combine part of its genome with another coronavirus. So they can mix in an unusual way for viruses.
So they can sort of pick and choose from each other.
Yeah, exactly. Like you have a component there that is perfect for the receptor of
Maja,
humans.
I like your arms.
Exactly. Okay. And I take your nose, and then now I can.
Yeah. Now I can reach further because you got longer arms.
Yeah. Something like that.
That's crazy.
Yeah.
They're really amazing to some extent.
Yes. They are super fascinating.
Is that sort of why that you are like scratching the surface in it? Because it's like why you keep this research is because it's part of it also. And I think what is also super fun now is with these new methods where we can really study everything, like the whole ecosystem of microbes, because before you could only like study some bacteria that you could grow on a plate, or you could never say anything about the complexity of
gut microbiome, for example. But now we are starting to be able to answer those kind of, at least ask the questions. Answers still difficult, but like to see, okay, actually what is modulating most of the bacteria in our guts are different viruses, like phages. Phages are viruses that infect bacteria, and it's when the composition of bacteria changes, because viruses infect the bacteria and kill off certain and keep some, and like it's super interesting, I think, to look at this ecosystem of microbes, and also not only when they're doing disease, but like when they are actually good for us to some extent, and training our immune system in different ways.
Because that was going to be my question. Are they always bad, or are they actually essential for our survival?
No, exactly. They are not always bad. That's why it's so few that are actually causing disease, and it's just that we tend to focus on them because that's where we panic.
The more that we map viruses and understand their genetic code, their origin, and how they change over time.
Yeah.
Do you think that will make us in the future able to come up with treatments?
Yeah, definitely. Like this, I think the example with the coronavirus now is a very good example of that.
Yeah.
Like I'm trying always to tell people now, is if this would have happened 30 or 40 years ago, we would not know at the moment what it was. We wouldn't know the sequence of it, what it looked like. We could not diagnose it. We could definitely not find vaccines or make drugs. But now, after just like a few days, they had this full sequence of this virus, which is then using high throughput sequencing, like using exactly the method that I'm using.
Yeah.
And then you had the sequence, you could share it with the world quickly, and people could start isolating the virus, and we could make vaccines, and there's already like trials to try these new vaccines. So everything is moving in a speed that has never ever been seen before. And so definitely it will save many, many lives. Yeah. In Uganda, where we work on tick-borne diseases. So especially because ticks carry a lot of diseases, and they also do carry a lot of viruses, and the hypothesis there is that there would be definitely a lot of viruses that haven't been described that are carried by ticks and can also cause disease probably in animals and humans.
But the viruses, are they found inside the tick?
Yes.
And then when the tick put its little teeth in
its mouth parts in its saliva, so it's kind of...
So the viruses is then found in the saliva as well?
Yeah,
or
it comes with the saliva. Yeah, it comes with the saliva.
So even in saliva there's viruses?
Yes.
It's amazing.
And actually that's why one thing that can be interesting, or I think it's like, if you get the tick and you remove it immediately, you can be quite sure that you don't get bacteria and parasites from it, but the viruses you will get immediately because they are in the
spittle.
Exactly. Whereas for the bacteria and parasites to go over, they need to be kind of activated by the temperature of the blood that goes into the midgut, and then it circulates, and then the bacteria travels and goes to the salivary glands. So it takes usually 24 hours or even sometimes 72 hours for bacteria and parasites to cross over, but viruses go immediately. So it's like, to avoid Lyme disease, it's good to remove it immediately,
right away.
But to avoid tick-borne encephalitis, you have to take the vaccine, because even if you remove it immediately, you can. The virus can still have been.
They are amazing. That's
I can talk about another project also.
Yes, let's talk about another project. I really like it.
Yeah, because another project, it's on neurotropic viruses.
Yeah.
So this project is based here in Sweden. So we look at piglets that are born with a congenital tremor, so they are shaking when they are born.
Yeah.
And very often they cannot survive because they cannot suckle. And it's known that this is a virus that they get during gestation, or like the sow is during the pregnancy, some virus is infecting her, and then it crosses the placenta to the piglet, and they get the difficulty to, they get this tremor. And we've been looking now in Sweden to see what is causing this. And when we got the money, it wasn't known anywhere in the world what was causing it, but exactly the same year that the project was funded, one virus was found that could cause this disease. So it's called atypical porcine pestivirus, and now we've also seen it in Sweden.
Yeah.
So, and it seems to be very common in these piglets that have this disease. So.
Wow. But that one crosses over the placental barrier.
Yeah, exactly. very scary.
That's very scary.
Yes.
It's really fun.
Yeah, I enjoyed it. Thank you very much.
So I will leave some notes so people can find you if they have more questions, like the web page and stuff like that. And that'll be it. Bye.
Bye bye.